Då hälsar jag Ola välkommen hit för att fortsätta i Life Science och Healthcare Arena.
Okay.
Och jag kan också fortsätta att stå här.
Okay, yeah. So that's fine. So thanks so much for the invitation. It's a pleasure to be here and present actually a project that I care a lot about myself called the Open Facts. So I'm working in Mölndal, just south of Gothenburg, at the AstraZeneca R&D site there. So I'm part of what's called a research organization, which is kind of a bit actually separate from what Kerstin is, which is more the clinical development one. And of course, we have our own semantic data integration problem and issues for our specific data. So there I'm part of an organization called Discovery Sciences, where we are basically, it's a global organization or function, and it supports the early part of drug discovery. So I'm very much involved in the first steps of a drug discovery project, where you will find the molecule that is the starting point for a new drug. And then you need to work on it approximately for 10 years, then hopefully it comes to the market. If you go to the next slide. And first, a little bit to give the background the projects, where it was originating from. And that's kind of how we are currently in the pharmaceutical industry, where we've been during the last actually decade. There have been a lot of pressure on the sector, which has been reflected here in Sweden by Astra actually closing 2 of the 3 research sites. So the one in Lund and one in Södertälje are closed. So it's only we left in Mölndal. And there's been several reasons why there's been a lot of pressure on it, particularly to lower cost. The one is the patent expiry. For instance, there we have Nexium, which is actually a drug that was developed here in Mölndal. There's more generic competition. As soon as the patent expires, other company can manufacture a drug, and then the price drops 90 to 95%. And also the cost for R&D goes up continuously. You need to do potentially bigger clinical trials, and you need to do more safety studies. So all in all, it creates a lot of pressure on the R&D organization that we should work more efficiently. If you go to the next slide. And one way to try to actually lower the cost is to do more pre-competitively, to work with our actual competitors in what we define as the pre-competitive space, but we are doing things that we need to do anyway, but it's not really around creating the specific drug that we like to sell on the market. So, and part of the pre-competitive space is definitely in informatics. So there, all companies are using, I would say, 3 types of data. If you look at the ownership, you have the public data, you have the data that you have licensed from content providers, and you have the internal data. And everything needs to be integrated. And since public data, everybody's using it. So it would be good if you could work together there, because there's a lot of different heterogeneous public data. And if you can work together to do the integration, then we need to do it only once instead of 10 times, which will need to actually we can save some money. That is actually important for us. And we had an article of some colleagues of mine published an article in Nature Review Drug Discovery 2009 about how we can work more pre-competitively. And from that, if you go to the next slide. And so to say, another is that over the last decade, there actually quite much have happened in and around the data. Data has become more open. There are much more what is important for us, what's called bioactivity data. It's basically which molecules will interact with the protein. It's much more in the public domain. The standards, I would say they're still far from perfect, but they're getting better. And that helps, of course, also with the integration. And I think that a lot of the major data providers for us, particularly the EBI, which is European Bioinformatics Institute, also getting better to actually organize their own data, which makes it easier to integrate. But still, there have been no real efforts before OpenFacts to integrate data across all sources and across all types of providers. Go to next. And then what we have now in Europe, I think, which is very good, it was called the Innovative Medicines Initiative. So it was part of FP7, the European Union Research Framework. And it's basically to create a public-private partnership between pharmaceutical companies and academic groups. And it's focusing on key problems like the efficacy of drugs, the safety of drugs, but also quite a big knowledge management component. And in total, the IMI has a budget of 2 billion euro. We're talking actually quite a big money here. 1 billion committed from the pharmaceutical companies and 1 billion committed from the EU. And a project like OpenFacts, we have a budget around 20 million euro, I believe, which is a significant sum, but it's actually a very small part of the total sum. I'm also part in another IMI project called the European Lead Factor. We actually create a drug discovery infrastructure for the whole Europe. And there we have actually budget 200 million euro. So what actually happens in practice is that the pharma companies discuss what is relevant for them, and then they put out a call, and then different academic consortia apply to win the call, and there's a winner announced, and then the FPA companies, the pharma companies, and the academics form like a consortia, and the OpenFacts is one of those consortia. So we started in 2011, and the original consortium was for 3 years, so it was until 2014. We got 2 years prolongation, so we will continue until 2016. And that's basically to create what we call an open pharmacology space. So in the area of early drug discovery, really work together to integrate the data according to, the best available technologies, ontologies, and etc. So the consortia consists of 15 academic partners, 10 pharma company, and 6 SMEs, small and medium-sized enterprises. And then we have different work package or cluster around the technical build, the scientific drive, and the community. And what is very important for the informatics project, actually, how do you handle the sustainability of the project? So basically here we have the vision statement: integrate multiple research biomedical data resources into a single open and free access point. So here we are talking actually integrating data that's already in the public to them, not what we have internally at AstraZeneca or at any other pharma company. So since you are not working in drug discovery, you might not really understand the business questions here, but at least for me, they are very important. So that's basically business questions that I ask, and my colleagues here in GlaxoSmithKline, or my colleagues in Sanofi or Novartis or Johnson & Johnson also ask. So this is typical business questions that we would like to have to answer in our day-to-day work. And for that, you need a lot of different databases. Campbell is an important one which keeps information about the interactions between a compound and a protein. A drug bank that was also mentioned in K-Connect is another one. And you see you have 2 different colors here. You have the more grayish one, and then you have the more bluish one. The bluish is the commercial data that we also like to integrate with the public one. We started with the public one because that's actually a big task in itself, but we also now integrating some commercial data to show that it works as well. And then internally, we're working also to integrate our internal data. Here's an article we published in Drug Discovery today, and it's also basically the article is focusing on the research question. So it's really the whole project is driven by the need from the pharmaceutical companies, but also from all the actually drug discovery efforts going on in the academic community nowadays. I think 10 years ago, all drug discovery was in the pharma company or biotech companies. But I think today there's drug discovery centers at many universities. Sweden has one, which is shared between Karolinska and Uppsala. But there are several like in London or in Dundee and in Max Planck in Germany and so on. So it's also relevant questions, not only actually for the commercial companies, but also for more and more academic institutions. But it's really, we define the questions up front, and everything has been basically based on that, which databases we selected and how it was integrated. So if you look at the discovery platform, it's a cloud-based system provided by a company called OpenLink. As I said, it's guided by the business questions. And the one architectural decision we take, even though we're using semantic web technology for the integration, actually, let's say the interaction with the outer world is through an API. That was, we discussed that a lot in the tech committee. Decision that go actually from API and say not a Sparkle endpoint was that to make it easier to build application on top of the data resource, and it's also easier actually to control the queries through an API. But yeah, of course, you would probably have more flexibility with a Sparkle endpoint, but it's always a trade-off, and you need to make a decision. If you're more interested in how we set up the platform, there have been several articles published which are available, which has been written by the tech team, which has been led by the Vrije University in Amsterdam, but also a lot of contribution from University of Manchester in England. And I think in several talks already, there have been discussion around standards, and there's a reason for that, of course, because standards are difficult. It's quite easy to not be, you don't like the standards, and then you think you created your own universal standard, but actually there's just one standard in the row of standards. So we tried as much as possible to adapt to the standards that are available. For instance, we have not developed any own ontologies. We have shoehorned in our data in available ontologies, but we are based. It's based on Sparkle, RDF, SCOS, Void, and so on. QEDT for units, for instance. What is important for us is the provenance, because not all statements in the scientific literature are correct. Actually, there's actually quite a few scientific articles where you can't reproduce the results. So which means that you will always like to go back to the origin of the source. And so the provenance is important. We have used quite a lot what's called nano-publications there. So we have the provenance, because that's, I know when we do searches or informatics analysis, the provenance is always very important for us. And here's an interesting question, almost a philosophical question. So are these two molecules the same? And of course, there's no yes or no answer. It depends on the context. So actually, it's a very famous drug. It's Glivec, which is an oncology drug from Novartis. And here is basically the active component. And up there is it basically when you have it in the pill that you actually swallow, then you have it in a salt form. So it's a difference between the active ingredient and actually the whole pill? Are they the same or different when you want to do a search? And the question is, it depends. And that had made that also, which I will come to a little bit later, made also impacted the architectural decisions that we needed to take for the system. Okay, go to the next. And then if you go to the next, as an ecosystem. So I think this slide actually describes the system, how it's built. So you have a lot of data sources that are being transformed into RDF format. For chemistry, it's a bit different. You need actually what's called a chemistry registration system, because you would like to do some specific operation with chemical structures like substructure search that you can't do in a triple stop. But besides that, all databases get uploaded into what we call the data cache, which is the virtuoso triple stop from Interlink. But we don't link them statically, so we don't use like SCOS same as or something like that. But we resolve that in runtime, and that means that at runtime you can decide if those two molecules are equivalent or not, if the URIs should be considered as equivalent or not. And I think it's very powerful. It might be something that will not be relevant in many other fields, but I think for us that's very useful. And that's handled by the IMS, the Identifier Management Service. So there you decide if two URIs are actually equivalent or not. And then we have an IRS, also an Identity Resolution Service, because the user basically write the name of the protein that he thinks is the right one, which it usually isn't, because a protein can have very many different names. So you need basically to take the name that he or she writes and then check with vocabulary so you get the proper names with the proper URI. But this is basically the system, and then you have the API, and you can export like JSON or XML format. And then the idea is, this is really the core platform, and then you have applications sitting on top of the platform. This is just to show also that it's really a platform, and then if you would like to have access to the API, it's no problem. Anyone can sign up for free and start to run queries to check what they get out, and we are definitely encouraging people to build application on top of the data source. If we go to the next. So here's an example of applications that have been built on top. That's some simple, we have a simple browser that you put in a protein name, a compound name, you click on a button, and then the results come up. There are some more what we define as advanced analytics, and we show some screenshot later, where you can more manipulate and look at the data in several different ways. There's more predictions, so the data there, it's a lot, say, compound to target how strong they bind, and then you can build a model of the data there to predict new compounds, how strong they will bind to a target. And then I think what is also very important, at least in the pharma, is we work a lot with workflow. Not everyone that is working in informatics or in my team, computational chemistry, are actually that proficient in programming. So we have a lot of workflows where it's more you drag and drop it. So integration with that is important as well. Mm-hmm. So here just a screenshot to show one application called PharmPrep that was part of the Open PHACTS and developed by a group at the University of Barcelona. And it's basically, just to describe it very schematically, you can see which compounds that are active on different targets, and then you can compare them. If you're working with that type of application, I think it's a powerful tool. We are, as you say, we are not using this particular. We're using the methods, but we had a system already up before that we are using. So it's a powerful way of looking at molecules. The next is also a molecular browser developed by an SME in Germany, where you can look up compounds from the data through the API. You can find similar compounds based on structure, based on properties. You can import your own compounds and compare those compounds with the compounds in the database. So it's also a handy analysis tool. And I should say, this application have been developed mainly between academic group and SMEs with medium-sized pharmas. There's 10 pharmas in the company. I would say six or seven are pretty big, like Astra. So we have a lot of tools already internally, but there are some smaller pharmas as well which don't have internal tooling. And for those, these type of applications are very handy. If you go to the next. And this is an application from University of Manchester. We'll have a product called Utopia, which you can use to annotate files and PDF documents. And here they have implemented open facts. So if there is a compound name in the document, you can get the structure from open facts. I think it's a very handy way if you are looking for an article, you see a name of a compound, but you don't know actually how it looks like. You just hover over and then the structure comes up. So that's also an application. So here we have workflows. And the Open Facts platform have been integrated in 2 workflows. The one below is called Pipeline Pilot. I think that's a workflow that is now, it was Engkvist before it's with Biovia. And that's a commercial product, and it's pretty common that we are using that in the pharma industry. The one above is called NIME, so that's an open source workflow that was, I think, originally for chem and bioinformatics, but now it's more like a business intelligence analysis workflow tool. But both have nodes for open facts. It's very easy to create your own workflows where there are nodes included that is collecting data from the Open Facts platform. And then we have the sustainability, which is actually, I think it's one of the important points in basically any EU project, actually what happens afterwards, because you have a lot of money only for a short time. And what do you do with all the data you have generated afterwards? This is Carol Goebel, who's professor of computer science in University of Manchester, and she was a bit philosophical, and yes, and she concluded that we need money for maintenance, which I think is true. And if we go to the next. So what we created is an Open Facts Foundation, which is part of what Phil mentioned, the Big Data Europe. So it's a not-for-profit membership organization registered in the UK, and it will take over the Open Facts discovery platform and get subscribing members. And quite a few farmers have signed up already, but I think probably we need some more. And it's basically to sustain the platform, to extend it, to bring in more data sources, and yeah, to keep the momentum going. And it's an independent entity, so it can be part also of European Union proposals like the big data. So hopefully that will also be a stream of revenue. We hope it will take off. There have been similar initiatives earlier in pharma creating not-for-profit organization that have worked out. So we are definitely hoping that this one will as well. Just to end with a few slides how we're actually working with it internally. So if you look at this slide, so that's what we have the API from Open Facts, and basically what we have done, we have extracted all the data from the platform and put it in basically a type of triple store internally called the Cortellis data fusion. It's nowadays a product from Thomson Reuters. It was an independent biotech company called Engkvist in Boston before, when we started our business relationship with them. And up there, it's what's called a mass spectrometer. So we are doing a lot of proteomics experiment where I'm sitting in an organization, particularly what's called chemoproteomics. So we get out proteome data that is potentially very important for us, because we have a molecule, but we don't really know which protein it interacts with. So we get out the experimental data, and then we analyze that together with the open FACTS data in the Cortellis data fusion. And we're using a lot of visualization called a triple map, which is part of that platform, which I think is very powerful, where you can walk around along the different triplets, how they connect to each other. It's Tom Plasterer, which is a driving force for linked data, and a colleague of Kerstin that has been building that system with some assistance from me and also from my colleague that was calling the chemical biology team over in Boston. And we've got one more slide. And then basically another application which has been pioneered by a postdoc that was working with me, Linda, and now been taken over by Peter, who's sitting over there. And it's related to how we find new starting points for drug discovery projects. Usually what we do is a high throughput screen. So we have a substance collection of 2 million compound. And for an important project, we screen them in the lab, all of them. Of course, it's not a person that is doing that, it's a robot. But nevertheless, it generated quite a lot of data. So it's important that you analyze that in a very good way. And what we have used there, it was called the bioassay ontology, which is an ontology to describe basically how you set up your system when you measure when a compound interacts with a protein. And then we have annotated all the historical high throughput screen from the last 10 to 15 years. So it's around 500 to 600, mainly done by Linda and some summer worker helping Linda. And then it's put in a triple store, which is known as Legograph, and which Peter is working with. And then there we have a Sparkle endpoint, and we have an interface, so people can see which screens have been run before that is similar to this screen. And what we would like to identify is if a compound, when you screen a compound, it's a signal that's active. It might depend on two things. It's actually an active compound, but it also might be interfering with the technology that you're using when you're screening. And those more what's called false positive or frequent hitter you want to remove. So we're using basically our, so to say, data, screening data annotated with the bioassay ontology and querying the triple store to find out which compounds are true hitters that you would like to progress, or which compounds that are what's called false positive and you should remove and not continue in the project. So that's Linda and Peter, and also Isabella Fireby in the team has been involved there. If you go to the next. So this is the acknowledgement. So it's a big project, it's a complex project, but I think we have made a lot of progress in semantic integration. It has been definitely, I think, for all pharma companies, a learning journey. In drug discovery, I think there was basically no knowledge about semantic technologies 2010, but we have really learned a lot, and it's been actually a really pleasure to be involved in the project and to work with a lot of the leading, so to say, academic groups with semantic integration in Europe, and also with colleagues in the other pharma companies, and particular thanks to GSK or Stefan Seng, and the other has been led it from the industry. So he has been the chair from the industry, and I'm the deputy chair. and then Bryn and Bar and Mons from the academic who has been in the executive committee. Okay, thank you.
Thank you. Any questions? Well, I thought it was a very good presentation, and I had a few thoughts, as you said. I mean, reflecting back to what Phil said earlier, sometimes you already know the questions, which is why when you lean towards using RESTful APIs, very simple APIs, rather than going for the complex Sparkle queries, which could become very complex.
It can.
Yeah, depending on your corpus and your material, and your material is complex from the starting point. So I thought that was a very slick way to addressing this. Just making a RESTful API with some of the most common threaded questions that you will see. Was that a big issue when you were, because if we look at the different examples you had going inside, I mean, you've been doing this project outside your company, but working with, obviously in Mölndal, but quite a lot in your international group of people here. To be a kind of a speaking person for these semantic things inside, as you said, I mean, you already have loads and applications and databases, and the resistance from, well, we already have something that's similar to this. What's your thought?
It is a challenge, and I think Kerstin is nodding a bit as well there. It is a challenge. It is a new technology. It will change a bit how we are working. So it's a challenge, but I think you need to be a core that is really believing it, and then you need to for the big masses, at least in the beginning, hide the complexity.
Yes.
So either through an API or through the triple map browser that they have in Cortellis data fusion. So people are working there. So they're actually not really aware of what's going on under the hood.
That's good.
Yeah.
And I thought that was a very good example of just simplify for the end user community who doesn't care whatsoever about semantic web, just wanting use cases where their practical work actually works better. And for the ones who do in the integration points, but just make it simple as JSON or APIs or whatever to make do all the things. I think that's two key takeaways from me anyway. So thank you very much.